All righty. Today is Thursday, September 9th, 2021. The schedule always gets scary when it starts getting short like this. We only have a little bit of material left to cover today, finishing up our discussion of blood and blood typing. And then, like I said, we should have plenty of time for a question and answer review, all leading up to Tuesday's exams. Again, remember both exams are timed. But more importantly, both exams must be completed during class time. What this means is if the lecture exam, for instance, has 2 hours to complete, but you wait till 4 o'clock to start it, you're only getting 35 minutes to be able to complete that. So again, the lab exam, I haven't finished doing these lab exams, probably going to be somewhere about an hour and a half. Lecture exam will probably be somewhere about 2.5 hours. So that's 4 hours of exams, easy to finish that into your 4.5 hours of the class time. Again, you're not required, but I encourage you to start with the lab exams and to start right at the beginning of class. 90% of the issues people have are with the lab exam. Again, we're using the one question at a time format because hopefully that will help with loading speeds, but make sure you have good internet, make sure you're close to your router if you're wireless or even hardwired into it. Make sure you have enough RAM and enough working space on your computer. And again, when you're doing the checks, it should warn you about all that stuff. So I think we've talked about that, and we can talk about that more during the review today, but I just want to make sure to emphasize those things. And then lastly, I also want to make sure to emphasize, as we've talked about, it is always important to be here, but there is no time for an exam hangover. Thursday, we are diving into the new material. And if anything, I think the new material might be slightly, well, normally in the classroom, I think it's slightly more challenging because you've spent the first couple of weeks holding the models in your hand and looking at the hearts and looking at the blood vessels and looking at the arms and looking at the legs. Whereas the endocrine system and lymphatic system and immune response don't lend themselves to that. There aren't really as many things you can hold in your hands. But since we've been online, you haven't been able to hold things in your hand. It might actually be hopefully a little bit easier to comprehend this information, dealing with it in this format. But we are forming groups for 50-point presentations. You're picking your topics. And starting the work. All on Thursday. So it is very, very important that you're here on Thursday. If you're not in here on Thursday, you're not going to end up in a group. You're not going to have a topic. You're not going to be able to start working on this material. And as a result of that, you are going to get a 0 out of 50 on this assignment. And that is something that I promise you, you do not want to do. So make sure you are here Thursday for that. All right. Questions on any of that? Like I said, I think that game plan is pretty simple and straightforward. Like I said, there isn't that much left to do. So let's pick up the lecture where we left off. One second here. All righty. So we left off last time and we had just finished talking about this idea of these pluripotent stem cells found in the red bone marrow that are going to be responsible for producing all of the formed elements associated with the blood. And as we mentioned, that generic process of forming all of the formed elements of the blood is what we call hematopoiesis, or sometimes also referred to as hemopoiesis. And so that for every single formed element that we're going to be dealing with, you need to know both the primary and the secondary stem cells that are responsible for them. However, as we also talked about, that isn't particularly challenging information, because how many primary stem cells are there for all of our formed elements?
One.
Exactly, just that hemocytoblast. So really, if you see that question on the exam, you don't even have to bother looking at the picture. You know the answer to it. And how many secondary stem cells are there?
Two,
right. The lymphoid, which just make the lymphocytes, and the myeloid that make basically everything else. Now, in the last class, I mentioned at the end, but we hadn't had a chance to talk about it yet, that the one pathway you would be responsible for was the pathway involving the formation specifically of our red blood cells. And that process specifically of forming our red blood cells is what is called erythropoiesis. As we have already talked about, for our red blood cells, we know the primary stem cell. And what is that primary stem cell again?
hemocytoblast.
Excellent. And what is the secondary?
Myeloid.
Excellent. Now, remember, this myeloid stem cell is also a pluripotent stem cell. What does that mean?
Can make a range of different cells.
Excellent. When it divides, it is capable of producing multiple, more than two, a small subset of cells. So still at this point of a myeloid, it is not necessarily 100% guaranteed to become a red blood cell. However, one of the things that our myeloid stem cell can become is a proerythroblast. And this proerythroblast is our committed cell. And what, of course, do you think we mean by a committed cell?
That it's going to travel down and become a erythrocyte.
Yeah, at this point, as a committed cell, there is only one thing it can become.
So would you say it's specialized?
It honestly, no. At this point, really what it is a unipotent stem cell. It is going to divide to produce multiple cells, but all the cells that it produces are going to become red blood cells. But I like where you're going, and I like where you're thinking with this, because it is, again, one of the keys to a definition of a stem cell is it really doesn't have any other job. So if we were talking about the cell becoming specialized, where now it has a specific function, that proerythroblast divides to produce what we call early erythroblasts. And this early erythroblast now has a function. Notice if we look at the pretty picture of this, again, as we always think about, color is one of those things we don't usually spend too much time paying attention to when we're looking under the microscope, because when we do that, often the color is determined by the stain. But one of the nice things about when we're putting blood on a slide, we don't have to necessarily put stain on it. So when we're looking at these cells under the microscope, as you can see here, they have a massive nucleus, obviously something that is very, very different from what we expect in a normal red blood cell. And it has a very dark purple color. If I remember correctly, if I think back to 430, when I remember looking at the neurons under the microscope, they also had a very dark purple color. And why did they have that dark purple color? Does anybody remember? It's okay, I wasn't paying attention either.
Ribosomes.
Exactly. Remember in our neurons, we had a fancy name for it. We called it Nissl bodies. But eventually, because it was the Nissl stain that made them show up. But eventually they realized that what they were calling the Nissl bodies were these flattened, interconnected membranous sacs with all of these fuzzy little bumps on the top of it. And so you kind of get the right idea with the ribosomes, but in fact, what it is indeed a massive amount of rough endoplasmic reticulum. And of course, what do we use rough endoplasmic reticulum for?
Produce proteins.
Produce proteins. And what protein do you think we're going to be making an excess of here?
Hemoglobin.
Exactly. The cell begins to make massive amounts of hemoglobin. Notice as it continues to make more and more hemoglobin, the hemoglobin, it starts to bind oxygen. And as it starts to bind oxygen, the cell starts to take on a more pinkish color. As I've said many times in histology, when we're looking at it, color doesn't matter, except when it does. And this happens to be one of these cases when it does. So normally, so as you see, as it moves from a early erythroblast to a late erythroblast to ultimately a stage called a normoblast, the cell is getting more and more pink in color as it fills up with more and more hemoglobin. And that's what brings us to this normoblast stage. At the normoblast stage, hemoglobin production has become so high and there's so much hemoglobin present that the cell basically decides, you know what? I don't need this pesky nucleus and all these organelles anymore. So at this point, as a normoblast, the cell actually ejects the nucleus. And not only does it eject the nucleus, but it also ejects a lot of its other organelles as well. At that point, it basically becomes a big round bag of hemoglobin. And of course, what do we call this big round bag of hemoglobin?
Erythrocyte.
Are erythrocytes big round bags? Or are they biconcave bags?
The reticulocyte.
Exactly. It's not a erythrocyte yet. It actually, as a big round bag of hemoglobin, we actually call it a reticulocyte. A reticulocyte basically is what we consider an immature erythrocyte. But it is actually as a reticulocyte that it is actually released from the bone marrow. Into, oops, the blood vessels. And how might it get into those blood vessels?
Capillary.
Yeah, but we can probably do a little better than that, right?
The venous side of the capillary.
I'm sorry?
The venous. side of the
Okay, so
capillary?
I see where you're going with there, because that is the side where reabsorption. I was thinking more in terms of what specific type of capillary. You're not wrong, but I was thinking in terms of what type of capillary would have large enough openings where a small reticulocyte would be able to get into the capillary.
Is it sigmoid?
Was it sigmoid? It's close. Did start with an S.
Hmm.
I can wait.
Sinusoid?
Excellent.
Is it-
So via those sinusoid capillaries, those reticulocytes are released into our blood. After about a day or two, they then mature into erythrocytes. So that is our developmental pathway of a red blood cell, this process of erythropoiesis.
So if the blood travels through the whole body multiple times, does that mean it'll just like go into the lungs, or will it take another path, or
The
well, after it goes to the
reticulocyte?
lungs a couple times? Yeah.
Yeah, sure, absolutely. It can go anywhere in the body. And in fact, at any one time, I think it's somewhere like, I think it's like 0.5 to 2.5, somewhere like that percent. Maybe it's less than that. 0.5 to 1.5, something along those lines. I think I have it written here in a second. Of your blood cells in your blood right now are reticulocytes. There's something like 2 million a second, or some crazy number of these are being released into your blood supply. And like I said, somewhere around 1 to 2% of your red blood cells are actually immature reticulocytes. And that's actually an important number, right? If you have just had a major injury or a major surgery, what would you expect that number to be, higher or lower?
Lower. Actually, no, it'd be higher because it's got to produce more.
Right, you're producing more blood to replace the blood that you've lost, so it's higher. So, a day or two after surgery, that should be high. If a week after surgery, that number is still high, is that something you might want to worry about?
Yeah, maybe some internal bleeding.
Yeah, exactly. That could be an indication. So it's actually one of the things that they will monitor in those types of cases, to look for internal bleeding. Absolutely. So I've done it on the pretty words. Again, we did that.
So
We did
Professor, is there not like a magical moment where it turns into an erythrocyte, but is there kind of like I've gone around a couple times, let me turn into a red blood cell now?
It's a great question. I do not know the mechanism by which it essentially changes its shape, is really what's happening, going into that biconcave shape. But I'm not actually aware of how that occurs. It's a great question. I don't have a good answer to. I just know that it takes about a day or two for it to mature into that distinct and unique shape.
Can it be just protein denaturation, a change of shape?
Well, but in this case, we don't want to denature the hemoglobin. We don't want it to be non-functional. Really, what's changing is the shape of the plasma membrane. So I don't know what it is about the plasma membrane that changes, that causes it to narrow its shape. Maybe it's something about how as the hemoglobin becomes oxygenated, maybe it changes some characteristic that causes it to change the shape. But I honestly don't know. It's a great question. I don't have a good answer for you. Someone might know, but I'm afraid I'm not one of them. Although you have me curious now, I may look into it. All right. Questions on this developmental process? All right, excellent. So now we know how to make more erythrocytes. But of course, as we've talked about, we need to be able to control that mechanism. And since the whole point of the erythrocytes is to carry oxygen, again, I can't emphasize this enough, 98.5% of all oxygen is carried by the red blood cells. So one of the things that we want to do is measure the oxygen carrying capacity of the blood. And if the blood isn't able to give the tissues the oxygen they need, we consider that a state of hypoxia. And as we've talked about, numerous things can cause this. It could be a problem with anemia, like we talked about, where you don't have enough red blood cells, or your red blood cells don't have enough hemoglobin, not enough iron in them, right? It could be circulatory problems where, like we talked about, heart failure or congestive heart failure or something else, blockage. We talked about high altitudes where there's less oxygen in the air you breathe in. It could be the plastic bag someone's holding over your head, right? For whatever reason, we need to monitor how much oxygen the body is able, the blood is able to carry. And where would a good place to do that be? Where is a good place for you to vitally get a blood to it at all times, a large amount of blood to it at all times?
Low altitude?
Well, true. Yeah, absolutely. We will get more oxygen at low altitude, but what part of your body would be a good place to monitor the oxygen carrying capacity of the blood? What part of your body, what organ in your body gets a massive amount of blood all the time?
Brain.
Your heart?
Brain would be good. Heart would be good, although those are pretty busy doing other things. What's really interested in not just the blood, but what's going on in the blood?
Kidneys?
Kidneys. Excellent. Kidneys. So in our kidneys, we have a chemoreceptor that measures the amount of oxygen in the blood. And if the oxygen level drops, our kidney releases a very special hormone called erythropoietin. Right. Again, if you follow any kind of endurance sport, like bicycling or something like that, you may have heard before about someone getting caught doping with EPO. Right, and EPO is erythropoietin. EPO, erythropoietin, basically does two things. The first thing that it does is that it stimulates the hemocytoblasts to become, let's say it this way. It stimulates the hemocytoblasts to become myeloid stem cells, and the myeloid to become proerythroblasts. So essentially what we're doing here is we make more red blood cells. But the second thing that it causes the cells to do is it stimulates the cells to mature faster, to reach that reticulocyte faster, to make hemoglobin faster. So not only do we make more red blood cells, but we make those more red blood cells faster. So not only do we overall get more red blood cells, but we start making them faster than we were making them before. So we make more cells and our proerythroblasts develop into reticulocytes faster. All right, questions on that? And here's a pretty picture from a textbook showing that balancing act. I'm sorry, did someone have a question?
No, I just said no.
Oh, okay, perfect. All righty. I always worry that I speak over people. So that is everything we need to talk about for our red blood cells. Let's switch gears now and talk about our white blood cells, or as we mentioned, our leukocytes. Remember, vast quantities of red blood cells in a single drop of blood, whereas a single drop of blood only has about 5,000 to 10,000 leukocytes, a significantly smaller number. Remember also, as we talked about, these are what we could think of as true cells. They have their nuclei, they have their organelles, they can make proteins, they can divide, they can metabolize, they can respond to their environment, they can do all of those types of things. Now, 5,000 to 10,000 don't sound like a whole heck of a lot, since we know that these are going to be responsible for our body's defenses. But part of the reasons for that is that's, I should say, 5 to 10,000 in a healthy person. Because in a healthy person, only about 2% of your leukocytes are actually in circulation. The rest are outside of the cardiovascular system, right? Again, we don't think of red blood cells that way. The red blood cells don't leave the blood vessels to take a holiday in the pancreas and then come back. But our leukocytes are actually able to migrate into and out of the blood vessels. And so as a result of that, they can be found in our lymphatic fluid or our lymphatic vessels, lymph nodes and spleen. There are some found in our skin, lungs, and lots of other locations, our liver, other places in the body as well. As I mentioned, there are 5 main types. And those 5 main types fall under 2 categories. The first categories are granular or granulocytes. These are neutrophils, eosinophils, and basophils. And as hopefully you can clearly see from the pictures, what makes them granulocytes is they have a large number of vesicles. In their cytoplasm. And so these not large number of vesicles are what cause them to have this granular appearance to them. Neutrophils by far are the most common. Of all of our leukocytes. Notice how many nuclei does this neutrophil have?
4.
It does kind of look like 4. It turns out it's really just 1. This is a single multi-lobed nuclei. In fact, if you notice, all of our granulocytes have multi-lobed nuclei. So each of them, even though it doesn't appear that way, each of them just has 1 nucleus, but that nucleus has multiple lobes to it. Neutrophils are easy to identify because they have a relatively clear cytoplasm. They typically have the, wow, that's not even close. They tend to have the smallest of the granules inside of them, making it easier to see the cytoplasm and easier to see the nuclei. So to often the nuclei are most clear in a neutrophil. I don't have a good answer for you. I don't know why. Again, like I said, that brings us back to that whole problem of why. While most of our leukocytes have some phagolytic characteristic to them, neutrophils are our first responders when things are when we are damaged, when there's an injury, and once they leave the blood, they are activated, become phagocytes. Now, someone remind me again what a phagocyte is?
The cleanup.
I like that, but there you go. They eat
Yeah.
things. Their job is to find that pathogen, find that dead or damaged tissue, engulf it within them, and use their digestive enzymes from their lysosomes to break them down in bits and pieces. We'll actually talk about the mechanism by which they do that in the next section when we move to our immune response. So for right now, we just need to know that their job is to be phagocytes. Their job is to eat things that are bad, things that are abnormal, things that are not us, and then show them off. Right? Like a cat, it kills the bird and then brings it, it lays it at your feet so that you know. That's kind of what our phagocytes do as well. Notice our eosinophil also has a multi-lobed nucleus, also has a massive number of vesicles. But what's super distinct about this eosinophil?
Orange.
Yeah, the color. Again, color isn't important except when it is. Eosinophils are most easily identified by their very bright red, even sometimes brick red color to their vesicles. Eosinophils also help in our immune response. Of course, all of our leukocytes do, but they're primarily responsible in helping in the breakdown and digestion of, in particular, things like parasitic worms and stuff along those lines. So things a little bit bigger than viruses and bacteria. Notice this third one, the basophil, also has big, massive, and dark vesicles. In fact, there's so many, it's so dark, it's so distinct that we really can't see the nuclei. You're going to have to assume it has a multi-lobe nuclei because I'm telling you, but can you tell that by looking at the picture? Anybody see the lobes of the nuclei in there?
Very
Yeah.
faintly.
Yeah, maybe, absolutely. Anyone happen to know what the big dark granules found here inside of our basophil contain? It's okay if you don't, I was just curious. Histamine. What does histamine do for us?
It facilitates a inflammatory response.
Exactly. Basically, histamine causes inflammation. You're absolutely right. It basically does that by two ways. It dilates blood vessels to bring more blood to the area, and it makes the capillaries more leaky. So if you think about it, more blood rushes to the area, making the area red. And as we know, blood is warm, so it makes the area warm, red and warm. And by making the capillaries more leaky, more fluid leaves the blood vessel, causing swelling in the area. And as that swelling swells in the area, it pushes on the nerves, causing pain. Right? Redness, heat, swelling, pain. Those are the four cardinal symptoms associated with inflammation. And histamine is the cause. Basophils in our blood are a cell that have histamine, contain histamine, and release a lot of it. Didn't we learn about a cell in 432, really, in 430 that also contain histamine? Anybody remember the 430 cells that also contain histamine?
It wasn't.
Oh, there was the one in the skin.
Yep, one was in the skin, and the other, I'll remind you, is primarily found in the areolar connective tissue. So anybody remember the name of the cell in the skin that produces and releases histamine?
I think the one in the areolar, was it the mast cell?
Exactly, it was. So what was the one in the skin? Anyone remember? So when you scratch yourself, or you get hives, or you get the oil from poison ivy, and you get that rash as a result, that is due to dendritic cells located in the surface, in the epidermis of the skin. So you should have learned that in 430. So all of these are cells that have histamine, and all of these can cause that swelling, that inflammation, that edema.
Professor?
Yes.
Is there a specific number of lobes of nucleus, or is it always 4 for neutrophils and 2 for?
No, it's totally random. And like I said, I think it was, yeah, Samuel asked, it's a great question. I do not know why the lobes, why the nuclei are multi-lobed. I'm not aware. I don't think anybody that I've read knows why, but absolutely, it's not standard. It's not like the eosinophils always have 2 and neutrophils always have 4. It's just random. They always have more than 1. That's the one thing you can be certain of. That helps because that is different for our 2 agranulocytes. They are the monocytes and our lymphocytes. Monocytes are by far the largest of our leukocytes, and there are big eaters. They are a little slower to, excuse me, they're a little slower to activate. Typically, when you first have an injury, they're not going to be present. But if you have some kind of chronic injury or chronic illness, these slow giants will eventually wake up and migrate to the area, and they become what we call our macrophages, our big eaters. And our lymphocytes can be easy to identify because, as you can notice, they're almost all nucleus. Wow, that doesn't show up at all. They're almost all nucleus, a big, huge, massive singular nucleus and a tiny bit of cytoplasm. And these lymphocytes, which remember are the only ones that are made from the lymphoid stem cells, and these are the ones that are responsible for our immune response. Oops. Like I said, we'll go over these things in more detail when we get to second section. We talk about our immune response, but we need to know these things now. And as I've hinted at, do we have an equal number of all of these?
No.
No.
Neutrophils are by far the most common. And if you need to remember the order, just remember to never let monkeys eat bananas. And you will remember your leukocytes from the most common, neutrophils, to the basophils, the least common. I hinted at this in the last class. Notice I love this picture. I guarantee this picture is on your lab exam, although I also guarantee it's not the only picture on your lab exam that shows these things. But what I really like about this particular picture is that it has all of our formed elements on it. We can start easy. That's horrible. What is this formed element?
Erythrocytes.
Excellent. Oops. What did we say that one was?
Thrombocyte.
What about this one?
Leukocyte.
Well, but we want to be more specific.
Neutrophil.
Excellent. What's this one?
Lymphocyte.
Excellent. What's this one?
Monocyte.
Yeah, notice it's got that kind of U-shaped nucleus, not quite multi-lobe, but kind of bent over on itself. And by far it is the largest. This one?
Neutrophils.
Yep. This one?
Eosinophils.
And
Basophil.
there you go. And remember, for each of these, you need to know their functions as well as their names. And then for each of these, you need to know their primary and their secondary stem cells. All right, questions on that? All right, here are the developmental pathways for our leukocytes. And again, remember, as I mentioned, I'm not going to hold you responsible for the intermediate pathways. You don't have to worry about that information. You just need to be aware of the fact that our myeloid stem cells are the ones that are produced, the eosinophils, the basophils, the neutrophils, and the monocytes, whereas the lymphoid stem cell is essentially, if you think about it, this one here already, even though it's our secondary stem cell, at this point, it is a committed cell. Because at this point, there's only one thing it can become. This lymphoid stem cell can produce and only will produce our lymphocytes. So we did that. All right. And of course, what determines which of these are made?
Hormones.
Yeah, hormones and other chemical signals. A lot of these, so, and again, this is another big thing we will talk about in the next section. Typically, when we think of hormones, we think of hormones that get into the blood and circulate everywhere. But there are also a large number of essentially what we'll call right now local hormones. And so these local hormones of the bone marrow, growth factors and interleukins and things along those lines play an important role in directing these cells to become which specific type of white blood cell we need, which is important, right? From a diagnostic standpoint, when you take somebody's blood, there is an expected white blood cell count, how many of each type that you have. And if instead, when you looked at someone's blood, you saw they had a massive amount of eosinophils, what might that be an indication of for you?
Infection.
Or more specifically, not just an infection with eosinophils, but typically some type of parasite, like a parasitic worm. Yeah, parasitic infection. Excellent. Perfect. Off the page, Samuel. Excellent. Thank you. All right. Like I said, we will talk more about our white blood cells and their functions in the next section. But when we talk about our immune response, but that's enough information for that for now. The last thing we need to talk about for our formed elements are our thrombocytes, the platelets. It is a little bit early, but this is a good natural stopping point because from here, we're going to kind of go into hemostasis. So I think this is a good natural stopping point for our first break. So let's go ahead and take our first break. It is 12:54. So that means let's come back at 1:09, and at 1:09, we will restart the lecture. And I will start the recording at that time. So any questions before we take our first break?
No.
All right, excellent. I'll see you guys in 15 minutes. Let us go ahead and get started. So, as I mentioned, we've talked about erythrocytes, we've talked about leukocytes, we are now on to thrombocytes. Remember, as we talked about, a thrombocyte, a platelet, is really just a piece of a cell. It doesn't have a nucleus, it doesn't have organelles, it is just a small fragment of plasma membrane with a large number of vesicles inside of them. Now, there are a fair number of them in a single drop of blood. So again, somewhere between 150,000 and 500,000 in a single drop of blood. But again, we're talking about little cell pieces. We've actually seen under a microscope how they look like little pieces of chewed up bubble gum sitting on the sidewalk. And while they are not true cells, they do come from a true cell. Again, I'm not holding you responsible for the intermediate steps in this path. But what I do want you to see about this path is what happens is we start out with a cell that undergoes mitosis, but never cytokinesis. Now, it sounds big and fancy, the kind of thing you could call grandma on the phone and tell her, hey, guess what, grandma, this cell undergoes mitosis, but never cytokinesis, and she'll be very impressed and send you 20 bucks in the mail. But what does it mean?
The nucleus divides, but the cell doesn't.
Exactly. Mitosis, remember, is the division of the nucleus. The cytokinesis is the process by which we divide the cytoplasm, where the cell physically divides into two. So notice this megakaryocyte has multiple nuclei. Inside of it as a single cell. And what might the advantage of having all those nuclei be? Why might it be helpful to have a lot of nuclei?
Transcription, synthesize more.
Exactly. You're absolutely correct, right? Nuclei contain the information for making proteins. So that means more nuclei means more transcription, means more proteins. Because after all, as we mentioned, these pieces that are going to get ripped off of it are going to contain a vast number of vesicles, a vast number of proteins that serve some important functions. Basically, let me see the next slide. Yeah, okay, perfect. Basically, what this cell does is this cell, this megakaryocyte, basically sits on top of the capillary. So we've got this big, huge, multi-nucleated cell that sits on top of a capillary. Again, I have to work on my analogies because normally we're sitting in the classroom right now. And I would mention, just like you could go stand on the side of Auburn Boulevard, right outside our campus. And if you were to stand right there on Auburn Boulevard on the sidewalk and stick your leg out into the road, what would happen?
You might get hit.
True, someone might come by and take that leg right off because people drive about 50 miles an hour down Auburn Boulevard right over by our campus. And that's exactly what would happen. And that's exactly what happens to this megakaryocyte. This megakaryocyte takes extensions of its plasma membrane and sticks it through those intercellular clefts containing the proteins that we need inside of it into that cell. And as that red blood cell comes screaming by in that capillary, it basically rips off that little piece. And now suddenly we have that thrombocyte now floating around inside the blood. So basically these megakaryocytes are constantly having their arms and legs ripped off as they're becoming those thrombocytes floating around inside of our blood. And of course, the function of those platelets is what?
Blood clotting.
Starting the process of stopping blood loss, a process we call hemostasis, so that when we have a cut, it ultimately leads to the stopping of the blood loss, the clotting, the scabbing, and ultimately the healing of that tissue as a result of that. Absolutely. We'll talk about this in a little bit of detail. We got the test on Tuesday. This is another great example of how your book does an excellent job talking about these processes, but we're not going to go into it that deep. If you're interested, knowledge is for knowledge's sake. I would encourage you to take a look at it, but we're just going to superficially go over that information and understand the steps of this hemostasis. And we'll do that in a second. But the last thing I want to, and again, emphasize this point that we've talked about, is again, all of this differentiation from that hemocytoblast into which specific formed element is going to occur is driven by hormones and other chemical signals. We've already talked a little bit about some of these. Erythropoietin was the classic example that we talked about before the break. It's produced by the kidneys, and it both makes more red blood cells and makes those red blood cells divide more rapidly, mature more rapidly. But there are other growth factors that are can influence our hemocytoblast to produce certain things. Thrombopoietin is produced by the liver and helps to stimulate more platelet formation. And then, as I kind of talked about in the last class, before the break, cytokines are a class of what we'll call local, not circulating, but local hormones that play a very important role in determining which type of white blood cells are produced. So this is a very important process for helping to maintain the overall health of the body and allowing us to respond to specific dangers or injuries in specific ways. Again, as I mentioned, these are classes, things like interleukins, things like colonizing stimulating factors, stuff like that. We'll talk about those a little bit more in our immune response, but also when you get to microbiology, or if you've taken microbiology, you guys will talk more about it there as well. So even in the next section, we won't go too much in depth of these chemical components, because like I said, I know you'll be getting a lot of that in micro. All right, questions on that? All right, so as I mentioned, the last thing I want to talk about in our quote unquote lecture part of this is this process of hemostasis. Hemostasis is the process by which we maintain blood in our body, right? Allows us to respond to injuries. The first step in this is what is known as the vascular phase. When a blood vessel, so if we were to, again, have a blood vessel here, when that blood vessel is cut, what happens is nervous signals from the damaged tissue cause the smooth muscle to constrict. And so we actually get, that's way too big, this constriction of the blood vessel at the site of the injury. This is what is known as a vascular spasm. Now, does that mean that if I were to lop off my right arm, it's going to be perfectly fine because all those blood vessels are going to constrict tightly and no blood's going to be lost as a result of that?
No.
No, of course not. If the pressure in the blood vessel is too large, then that can overcome that vascular spasm in the artery. So this is effective for small blood vessels. But again, it's not going to help us much if you cut your femoral artery. And is something like this going to last for a prolonged period of time?
No.
No, it only lasts for about 30 minutes. So that muscle will contract for a short period of time, but that in and of itself isn't going to be sufficient to stop the blood loss. Luckily, chemical signals released by the damaged tissue also attract our thrombocytes to the area. This is what is known as the platelet phase. This, if you remember way back in 430, was one of the few positive feedback processes we talked about in maintaining homeostasis. Remember, with a positive feedback process, our goal is to enhance the disturbance. So basically what happens is damaged tissue releases a chemical signal, and that chemical signal attracts platelets to those areas. Once the platelets get there, they attach to the damaged tissue. They expand, they release their own chemicals, and now we have a larger chemical signal, which attracts even more platelets to the area. They attach, they swell, they release their chemical signals, and even more chemical signals are in the blood, bringing even more platelets to the area. And they attach and they swell and they release. And each time it gets bigger and bigger and bigger and bigger until finally the area is completely congested with platelets. And that hopefully is sufficient to temporarily stop the blood loss. Now, again, a platelet plug isn't going to be sufficient for stopping blood loss in your femoral artery. All of these work better in smaller blood vessels, but it is an important intermediate stage. And while the chemicals that are released attract more platelets to the area, the chemicals that are released from those platelets also lead to the coagulation phase. The big key to the coagulation phase is where we are going to bring those water-soluble fibers that we talked about out of solution. Basically forming these big mesh nets. Back in ancient times, and by ancient times, I mean when I was in elementary school. So far, far before any of you were alive. But there became this big, huge thing because everybody loved to eat tuna for lunch. But these tuna fishermen were using these big, huge, massive nets to catch all the tuna to meet all the demand. And what was the problem with that? Anybody hear about that from ancient history classes?
Were they catching things other than tuna in the
Yeah,
nets?
and what was the big, adorable thing that was being caught that everybody cared about?
Dolphins, tuna,
Exactly.
turtles,
Dolphins.
something like that.
No, it was dolphins, absolutely, right? Turtles aren't cute enough, mermaids, nobody cares, but dolphins, God forbid we catch dolphins, absolutely. So there was a big, huge fervor, right? Everybody stopped buying tuna fish. Tuna fish had to change their fishing strategy, and then everybody was proudly, you know, dolphin free and everything like that. Yeah, because these big, huge mass mesh nets caught everything. And that's what we want here. We want to form a big, huge mass net that is going to catch everything and catch the red blood cells and catch the formed elements and catch the pathogens that are trying to get in and form this dense structure that then those fibers tighten and dehydrate and form the scab on top of the injury that protects us from the outside world and gives us that foundation to heal and grow underneath. So these are our three big phases for hemostasis. Like I said, your book does an amazing job of going into great detail on them. We will just talk about them briefly. As I mentioned in the vascular phase, as we see here, injury to the blood vessel primarily stimulates an electrical response. Yes, there is some chemicals that are released by this as well, but primarily it's the pain of those electrical response that causes the smooth muscle to contract. That contraction of the smooth muscle, what we call that vascular spasm, can help to, as we know, decreasing the radius of our blood vessel increases the resistance. And if we increase the resistance, we decrease flow. So we can decrease the amount of blood being lost by this injured tissue. Like I said, it's not going to help you with your femoral artery because again, even when it constricts a little bit, the pressure is so great in there, you're still going to lose a lot of blood. And even in the small blood vessels, it doesn't last forever. It only lasts for a short period of time. Luckily, we have those platelets that are going to help us. As I mentioned, they're basically these chunks of cytoplasm containing granules inside. And not just random granules. Again, that megakaryocyte wasn't just making random proteins. There are basically two main types of granules found in our platelets. The first are what are known as alpha granules. These are the ones that are going to attract more platelets to the area These are the ones that are responsible for that positive feedback mechanism to bring them back. And as we mentioned also, these platelets are going to stimulate the process of blood clotting. By starting the process of drawing those those fibers out of solution. So here in our alpha granules, basically we are containing the chemical signals that are going to drive the processes. Now, not surprisingly, these are complicated processes. And so we also have these beta granules, or what are known as dense granules, that are going to play an important role in helping in those two processes. They're going to contain energy molecules like ATP and even ADP. Don't forget, ADP is an energy molecule. It doesn't have the high energy bond between the second and third phosphates that ATP does. But remember, ADP still has two phosphates, two very negative things that don't like each other. So the bond between them is still a high energy bond. And so it still can be an energy molecule. Calcium, of course, makes cells do wonky things. And then we have lots of enzymes and other chemical components that are going to help to facilitate this process of getting more platelets to the area and getting that blood to clot. As I mentioned, these platelets, when they attach to the tissue, they expand. And as they expand, they release their contents. Now, again, I've got this word rupture here, but I always think of rupturing like the popping of a balloon, right? Obviously, if this platelet popped like a balloon, there wouldn't be enough structure yet left to maintain that platelet plug. So yes, it does release its contents, but it still maintains some integrity, which is how it's able to form that plug that is going to very temporarily, hopefully stop the loss of blood. Again, as we talked about, this is that positive feedback process. The platelets attach to the damaged tissue. They form that spikes, they form that swelling, they release that content, and that content causes more platelets to come to the area. Again, this is that positive feedback process, homeostasis. And again, this is going to be very efficient at forming a temporary seal for that minor damage, which then will allow our clot to form. So a lot of things have to occur for clotting. It is a elaborate cascade, let's say it this way, cascading process. What do I mean when I say it's a cascading process?
It triggers different things to happen.
Yeah, exactly. I always think of it in terms of a row of dominoes, right? You have a row of dominoes. You hit the first one, it hits the second, it hits the third, it hits the fourth, and down the line they go. One event leads to the next, leads to the next, leads to the next. Like I said, your book does a beautiful job of explaining this. Somewhere in here, I'll jump ahead. Here we go. You can see this elaborate process that is involved in this. And again, your book does a great job describing it. I am showing you this picture, but you're not responsible for all of the steps. All right. However, what I do want to mention about this are a couple things. First is, like I said, there are a lot of things necessary and involved in this process. Vitamin K, calcium, again, anytime the cell does wonky thing, we need calcium, enzymes, other platelets, chemicals, and chemicals released by the damaged tissues. And the other thing to remember is that there are two separate pathways in this process. There is an intrinsic and an extrinsic pathway. If we cheat and go ahead to this picture, while we don't really need to recognize anything else that is going on here, we can kind of see what we mean by this idea. Notice the extrinsic pathway is actually from essentially outside of the blood. When they say extrinsic here, what they're meaning is outside of the blood. So this is, for instance, chemicals released by the damaged tissue. Right? So the tissue cells that are traumatized expose it to the blood and release those factors. Whereas intrinsic basically are chemical signals that are occurring. Why is that not letting me do that? There we go. Primarily inside of the blood vessel. So this is the inside of the blood vessel. These are the chemicals released by the epithelium of the blood vessel, by the platelets, and by other things along those lines as well. Again, I don't need you or care for that whether or not you understand these processes, but the point of both of them is the same. The point of both pathways is to convert those water-soluble fibers into their insoluble form. And when they're no longer soluble in the water, they come out of solution. And when they come out of solution, they basically form this big, huge mesh net. This big, huge mesh net covers the injury, stops the blood loss, and then will dehydrate to form the scab. And that scab, as you know, forms that protective covering that is going to allow the tissue to heal underneath. So again, if you're interested in understanding all of the parts of the pathway, that's great, that's good for you. I'm not going to test you on it, but just understand that we are converting that water-soluble fibers into water-insoluble fiber to form this big, huge mesh net that is catching red blood cells, white blood cells, all sorts of other harmful, potentially pathogens and other stuff that can damage tissue, forming that mesh net, stopping the blood loss. And then, like I said, dehydrating to form that protective covering, that scab that will allow the tissue to heal. All right, questions on that? Like I said, I know I'm doing a lot of hand waving there, but that's because I'm not holding you responsible for a lot of that information. So I'm purposely not talking about a lot of details because I just want you to have the big overview of those and not worry too much about specifics. All right. So questions on that?
I have a kind of a late question.
Sure.
The megakaryocytes, do they have like their own function before they break off?
Their function is to make the proteins that are going to fill the vesicles that are going to be released with the thrombocytes. So basically their job is to make more chemicals, make more vesicles, make more plasma membrane, because unlike you standing on the side of the road in Auburn Boulevard where your arm gets ripped off and you don't get to grow it back, they can grow it back. So they keep getting their arm ripped off again and again and again and again. And so they need to keep filling it with that good stuff to make all the thrombocytes. Great question. So platelets also are attracted to the site of the injury Their goal is to deal with the loss of blood, right? To help to form that platelet plug, to form that clot, to form that seal. When I think of first responders, though, I think more in terms of helping us to deal with any harmful pathogens that may come in. So the neutrophils are going to go to there. Neutrophils don't care about whether or not you're losing blood. Neutrophils care about what type of harmful pathogens may have gotten in. So yes, both platelets and neutrophils are going to go immediately to a site of an injury, but for very, very different functions. Right? One is there to stop the blood loss. One is there to protect the body from anything harmful that could got in because our walls have been breached. Does that make sense?
To go back to the megakaryocyte,
Yep.
why are their arms being ripped off instead of, say, exocytosis? Is it a passive process?
Well, so great question. What I would say is, if you think about it, if we had this cell that was just sitting on the surface here and just releasing chemicals into the blood, those chemicals in the blood would be able to then diffuse everywhere they wanted to. Not only is this megakaryocyte making all of the proteins, but it's bundling them up into a plasma membrane. So all the chemical signals or all the nutrients or all the things that are needed for that hemostasis process are all together in one location.
Got it.
Yes, you are correct. Both the formation of a platelet plug and the formation of the blood clot serve the same function. If we have a injury to the blood vessel, then the quick, fast response is going to be for those platelets to come in and kind of attach and anchor themselves to the walls and attach to more and attach to more and clump together and form this big cluster of cells, or really cell pieces, big cluster of plasma membranes and fibers and proteins that are going to help to try to stop the blood loss. It can form a very temporary structure, and like I said, can be effective in small blood vessels, but it's not going to be sustainable for a longer period of time. So yes, that platelet plug can stop the initial loss of blood, but it's not going to have enough structure or integrity to it to be able to maintain it while it heals. So instead, we need to then trigger that clot formation. And the big difference with the clot formation is we are getting all of these fibers that are coming out of solution, forming this big, huge fibrous mass that is going to make it much harder for things to get out. And those fibers are going to condense on the outer surface, forming that tough fibrous scab that is going to form that protective covering while the tissue heals underneath. So you've got the right idea. Both are going to play a role in stopping the blood loss and ultimately leading to the healing of the tissue. But the clotting is going to be, the platelet plug is going to be a more short-term solution, a faster, more rapid, but short-term solution. Whereas that clot is going to have more structure, more integrity, and ultimately lead to the scab that will be able to stay there for the days or weeks that it takes for that healing to occur.
I have a question kind of on that topic.
Sure.
I might just not be understanding it correctly, but so obviously we've all scabbed like on the outside of our body, but if this injury was to like a blood vessel, like you said, would we dehydrate those fibers and scab internally also? Or this is outside?
You wouldn't. So great question. It will clot, but it won't necessarily scab. When I'm talking about scabbing, okay, I'm talking about dehydration occurs on the outer surface. But okay, that is one of the problems that can occur if you have damage to the inner lining of your blood vessel. Then one of the things that can cause clots to form in that region. As if you've had a major surgery where those blood vessels have been cut, where the blood vessel is healing, clots can occur. And as the blood goes by, can that clot break off and cause
Break off.
problems?
Yeah. Okay.
Yeah, absolutely.
Yes.
So one of the things that typically after surgery, or if you have damaged, arteriosclerosis or something along those lines, you may be on a blood thinner to help to discourage that clot formation from occurring.
Got it. Thank you so much.
Yeah, so it's never going to dry up and become a scab, but clots can form inside the blood vessels, and absolutely that's a bad thing. Great questions. Any others from you guys?
Yeah. During the vascular phase, when the smooth muscle contracts, what causes it to contract?
Primarily, it is a neural response. When the blood vessel is damaged and the surrounding tissue is damaged, the nerves in there are pain receptors in there that send a signal to your brain that it hurts, but it also sends a signal to the smooth muscle. And so when it sends that electrical signal to the smooth muscle, that triggers the smooth muscle just at that location where the injury is to constrict, to try to help to decrease the blood flow in that area.
Thank you.
So yeah, that response is typically more of a neural response. Are there chemicals involved in it? Sure, because as we know, after all, when the neuron releases, the neuron stimulates that area, it's releasing a neurotransmitter, the chemical signal that is stimulating it. So it's driven by neurons, but it can be enhanced by other chemicals. Awesome. Anything else on this? Cool. See, again, this is a fun topic, but I know we don't have as much time to go into it. So unfortunately, it's not quite as important to me. Because like I said, I know you'll be getting more of this in micro as well. All right. The last major concept we have to talk about in this class is our blood typing. We kind of already hinted at this, and this is actually going to be a great feed in to the things we're going to be talking about in the next section with our immune response. But as we've talked about, it is very, very important for us to be able to tell the difference between things that are us and things that are not us. All right. So to do that basically requires two main things. The first thing you need to do is you need to have some type of tags on your cell. The example I use in 430 when we first start talking about this idea is, again, you've got an exam on Tuesday and 5 days off. So that's the perfect time to go to Vegas for the weekend, right? And again, since you made this decision last minute, you've got your black rolling bag and you rush to the airport and you get your flight. But by the time you get to your boarding group, all the overheads are full. So they have to check your black bag. When you get to Vegas and you get to that luggage carousel, how many black bags come rolling off of that luggage carousel?
432.
Exactly, a gajillion of them, absolutely. So how do you know which one is yours?
Your giant pink bow on it.
Exactly. Yeah, you've seen my bag. I've got a huge One Direction sticker and a big pink bow on mine. Makes it very, very easy for me to identify, right? Everybody has those tags. And cells need the same thing. These tags come in the form of what are usually proteins, but can also be carbohydrates as well, known as antigens. And so antigens are the structures that are on the surface of the cell. They are the tags that allow us to recognize them. Now, on our red blood cells are somewhat unique in that they only have a few of these tags. And by a few, I mean about 30. Now, there are 27 of us sitting here in this classroom right now. What do you think the chances that there are 2 of us in here that have the exact same tags, all 30 of the exact same tags on their blood vessels as somebody else in this class?
Probably not.
Probably not. Does that mean that nobody in this class can give blood to anybody else?
No.
No. Do you think there's a chance that there's at least one person in this class who could give blood to at least one other person? Yeah, absolutely. And that's because while we have 30 tags on the cover, and again, I know 30 sounds like a lot, but a liver cell has close to 200, right? So 30 isn't actually a lot, but of those 30 that are on there, they have different levels of importance. And in particular for our red blood cells, there are only 2 that cause an extreme reaction. And you may not have thought of it in these terms, but you're absolutely aware of it. Because how many people here know their blood type?
I do.
Yeah.
What is it?
Yes.
A negative.
A negative. Excellent. Anybody else know theirs?
O positive.
I got an AB negative. I got an O positive.
O positive.
Another O positive. Anybody else? Willing to share? No O negatives out there willing to admit it? B positive. There you go. B negative and B positive. Excellent. Notice when we talk about blood types, there are 2 parts when you describe your blood type. These 2 parts refer to the 2 sets of antigens. One is what we call the ABO antigen. And then the other is what is known as the Rh factor. So since these are the 2 that cause an extreme a reaction, these are the 2 we talk about when we talk about our blood types. So again, you may not have thought about antigens before, but many of you know your blood type or at least have heard of a blood type before, and that's what they're talking about. Now, it's all great to know what your tags are, but we also need to be able to recognize other foreign, abnormal tags, tags that are not us. To do that, we need some free-floating proteins in our blood plasma. And again, even if you haven't thought of it in these terms, I'm pretty sure you're aware of it, especially since we talked about this earlier in this section. We don't commonly, they can anatomically be referred to as immunoglobulins. And what did we commonly call those immunoglobulins?
Antibodies.
Exactly. We will talk more in detail about antibodies when we get to the next section. But for right now, what you need to know is they're basically these Y-shaped proteins that have some type of binding site at the end. 2 binding sites on our Y-shaped protein that allow us to recognize something that doesn't belong. And so these free-floating proteins, these free-floating tag identifiers, are known as antibodies. Let's switch to the whiteboard. And let's see what this looks like. Actually, let's cheat. That there. And that way I give myself some writing room. All right, let's take a look at some erythrocytes. There's one. There's one. There's one. There's one. So, as I mentioned, for our red blood cells, there are 2 important antigens. The first, as I mentioned, was ABO. Basically, your red blood cells on the surface either have They have some combination. Let's actually make this a little smaller so it fits on here either. Of two specific proteins. Let's say for argument's sake, we'll make these all blue because why not? Let's say, for instance, we have these blue circles on the surface that are, our A antigens. So again, on the surface of the red blood cell, we have A antigens. How do you think we identify someone who has the A antigens on the surface of their red blood cells? Oops. Well, okay, not okay. What do we call somebody? What do we call somebody? We call their blood type A.
I bet.
So their blood type would be considered A. Makes sense. Conversely, someone could have the antigens on their surface, and let's make them triangles. Now, again, notice I'm drawing them as triangles and circles. Is that really what they look like?
No.
No, of course not. But again, it just shows that they're different, and that's really what we're caring about here. So notice on the surface, the red blood cells, we have the B antigen, and guess how we identify this person?
Type B.
Blood type B.
Type B.
See how easy this is? What about someone who has both circles and triangles on their surfaces? Guess I should have used different colors to make them more obviously different, but too late now.
AB.
Exactly. This person's blood type would be AB. And our last person has neither A nor B on their surface, and absolutely their blood type is O. Perfect. Absolutely correct. So notice that first part, as we mentioned, the ABO has a combination of two specific proteins, the A protein and the B protein. And whatever combination of those you have determines the first part of your blood type. So again, on the surface of the red blood cell, we have no antigens. This one on the surface of red blood cells, A and B. And their blood type. Really? I've never heard that before.
Yes, I think it's in Europe. The first one is O, second is A, third is B, and the fourth one is AB.
That's kind of cool. Confusing, but kind of cool. You just have to remember the right order for them. But that kind of makes sense. The first and fourth makes sense. No antigens, all the antigens, but A and B is kind of arbitrary. I guess you put them in alphabetical order. But that's very cool. I had not heard of that before. That's awesome. All right. Antigens make sense. Yes or no?
Yes.
Yes.
All right, excellent. Perfect. Now, remember the other thing these cells need to be able to do is not only have their own tags on the surface, but cells must recognize tags that are not them. And again, remember, they're going to do that with antibodies in the plasma. So let's think about our first fella here, blood type A. What antigen is not him?
B.
B.
Excellent, B. So what he is not is B. So what that means is in his blood, he's going to need that specific antibody to recognize the B that's not him. So he's going to need the anti-B antibody. Remember, as I mentioned, antibodies are Y-shaped proteins that have a specific binding site on it. In this case, it would need to bind to B, so it's going to have a nice pointy binding site, allowing it to be able to grab onto it. Basically, when this antibody binds, it's a big warning flag. It's basically saying, hey, look at this. This thing over here doesn't belong. Somebody come and deal with it. And so that's what it's going to help us to do. It's going to help us to plant a flag on it to say, hey, this doesn't belong here. Come and do something about it. All right. So let's go with our second person here, blood type B. What type of antibodies in the blood plasma would they need?
A.
Yeah, an anti-A antibody. That, of course, would be a Y-shaped protein with a ball-shaped binding site on its surface to allow it to recognize anything that was A. Excellent. What antibodies would our person here with the AB blood type need?
Both anti-A and anti-B antibodies?
None.
So remember, the point of an antibody is to help us to recognize the antigens that are not us. Is A us?
Yeah.
Is B us?
Yes.
What's left?
Nothing.
Nothing. So notice in this case, they don't need any antibodies. They're able to recognize A as them, recognize B as them, so they don't have to worry. However, what about this poor fellow over here with blood type O?
Need both.
They're going to need both A antibodies and B antibodies, because anything that has an A on it, it needs to be able to recognize that as not belonging and do something about it. And anything that has a B on it, it has to recognize it as not us and be able to do something about it. There you go. Questions on our antibodies?
Professor, can you go into a bit more of explanation of why you can give anybody O?
That's actually a great question. Let's do that right now. Let's work our way through this. Let's think for each of these individual blood types. Let's think, what blood could this person receive? So of our blood types here, this person here with the type A blood. Now, again, the easy part is you can always get your own type of blood, right? So obviously this person can get blood type A because they're blood type A. Could we give them B?
No.
No.
No. Well, I've got a yes and I've got a no now. Which one?
No.
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 Let's
No.
think about it. This
No.
blood over here has the pointy antigens on it, and this has the antibody that will bind to it. So it'll recognize that as being foreign and not like it. What about AB? AB
No.
has the circle on it, so that should make it okay, right?
No.
It also has B on it.
But it also has the B on it, so it will recognize it as being foreign. But what about O?
Yeah,
Right.
because it has
O,
nothing to bind to.
exactly. O with that nice smooth surface doesn't have anything for the antibody to bind onto. So can the antibody tell that that O blood doesn't belong?
No.
No. And so notice that just answered your question. Because the O type doesn't have either A or B on its surface, it can basically be given to any of these four groups.
I thought the AB can receive blood from any type.
Well, we're working to that. Absolutely, we will do that too. But let's work our way up there. So based on what we just talked about, what do you think blood type B can receive?
B and O.
B and O.
You know,
Excellent. B and O. And as you correctly pointed out, they can always receive their own. We now know they can always receive O. Can AB receive A?
Yep.
yes.
Can A receive B?
Yep.
Yeah. So as you mentioned, if you have AB blood, you can basically receive all four types. And what can O receive?
Oh,
Oh,
Only O.
Excellent. Notice we could turn this around as well. Who can A give to?
A and AB.
A and AB.
A and AB. Who can B give to?
E and AB.
B and
B and
AB.
B and AB. Who can AB give to?
AB.
AB.
B only. And who can O give to?
Everyone.
All of them.
Excellent. Notice O is often referred to as the universal donor because they can give to everyone. And AB, I should say, is the universal receiver. You've got AB, you can pretty much pick blood up off the ground and stick it in your arm and be fine. Don't pick blood off the ground, but they could. All right. Questions on this?
Yeah, quick question, Professor.
Yes.
I could be wrong, but isn't O negative more of a universal donor, or?
You are 100% correct. And you're right. Notice we talked about how there's two important antigens, and we've only talked about one. So if we're comfortable with this so far, and it sounds like you guys followed along pretty well, let's take that next step where you're absolutely positively correct about. There is a second important antigen called that Rh factor. Now, unlike ABO, where there's two combinations of proteins, this is a single antigen that you either have or do not have. So if someone has it, and let's go ahead and draw this one, we'll draw this one as a square. So let's pick a different color this time. Let's go with brown. So you either have this antigen on your surface, or you do not. And if you have it on your surface, guess how we identify your blood type?
positive.
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 So this person up here is actually now a positive. This person down here is now a B, positive. And how would we refer to these two fellows over here on the right? negative. And negative. Excellent. Now, of course, as we know, if you either have or don't have an antigen, then that means you are either going to have the antigen, which means that it is you, or what else are you going to have?
An antibody?
So would our A positive here need an Rh antibody?
No.
No.
No. Would our AB positive need an Rh antibody?
No.
But what about this B negative guy?
Yep.
Yes.
So they need the anti-Rh antibody. So let's go ahead and draw that one. And what about our O negative fella here?
Yes.
Yep, so I'll cheat and sneak my antibody in here. So, yes, positive means that yes, you have the antigen. Negative means you don't have the antigen, so you need to be able to recognize it as something foreign. So for that, you would need the antibody.
But still they do have, let's say for B, B positive and P negative.
I'm
Right?
sorry, say again?
For the blood type, they still have the B positive and B negative.
Yeah, B positive and B negative people both have the B antigens on them. The difference between a B positive and a B negative, yeah, let's just do it this way, is that a B positive person would have both the B antigens on their surface and also the Rh.
And how do we know that?
That's an excellent question, and we will get there in a moment. Okay, I promise you we will do that information, but there's one more piece of business we have to deal with first, and then we will answer that question. Because notice now we can be a little bit more precise with what people can receive and what people can give. Since we've added that second layer, let's go ahead and do this again.
Sir, can I ask a quick question?
No. Yes, of course. Absolutely.
When we talk about like someone gives blood to another person,
Yep.
do we talk only about red blood cells or blood?
We're primarily red blood cells, because remember, as we talked about, 99% of the formed elements that you are giving someone when you're giving them a pint of your blood is red blood cells.
Because I think I both understand this and don't understand. For example, if O blood type O gives blood to blood type A, we're not talking only about the erythrocyte, right? It also we also give the plasma, which contains A antibody.
True,
So-
but okay, I see where you're going with this. Yes, the plasma of the individual who donated the blood will have some antibodies in them. However, the amount, as long as that person is healthy, which is one of the first questions you get asked before you donate blood, the amount of antibodies in their blood is going to be small enough where it won't cause a reaction. Right? We're talking about thousands of antibodies versus millions upon millions upon billions of cells. So it is the cells that make a difference. When you're donating blood, the antibodies in your blood have a negligible effect because there's so few of them compared to the number of red blood cells.
Okay, that makes sense. Thank you.
Yep. All right. So let's do this fully now. Let's come back up here to our A positive fella. What blood can A positive actually receive?
A
Actually, no.
positive and O positive.
Okay. Anything else?
O negative.
Anything else?
I believe that's it.
So let's think about this. If you have A positive blood, what antibodies do you have?
You could also be able to do A negative.
Negative, yeah.
Right. The only antibody you have is an antibody to B. So the only thing you can't take is something that has B on it. So notice if you're positive blood, you can get both positive and negative. But what about this fellow over here who's B negative? What can he receive?
Um,
B negative.
B negative, O negative.
Yep. Notice he has the antibody for the Rh factor. So if we tried to give him B positive, or if we tried to give him O positive, his antibody would bind it and cause problems. Let's flip it around. Who can A positive give to?
AB.
A positive and negative, and AB positive and negative.
Okay, so AB positive. You can always give to your own type, A positive. Can AB positive give to somebody who's negative? Can he give it to AB negative?
Yeah.
No.
No.
Oh no, it's the same thing because B negative can't get A, so yes,
Yeah,
you cannot do
AB
that.
negative. AB negative would have the Rh antibodies, so they would not be able to get it. So the only ones that can get it, the only people who can get positive blood are positive people.
I have a weird question.
Yes.
I one time heard that the Rh factor comes from the mom. Is that true?
I understand where you're getting that. Let's finish this and then we'll get exactly to that point.
Okay.
All right. You've got it's a great question and I will answer it, but let's just finish this real fast and then we can talk about that. All right. So let's do the same thing here. Who can B negative give to?
E negative.
AB negative.
AB negative. And AB positive.
So then B positive also.
That's correct. Exactly. Excellent.
Because, sorry, I just want to, so the B negative has the anti-Rh antibody, but the B positive doesn't have the Rh.
Okay, so I
you.
see you guys chugging down on this. And so let's see if we can write a rule here that will help us to understand it.
So it's pretty simple that negative type can receive only negative, but when it's given, it gives positive to and a negative, right?
Right, well, right, because here's what it depends about. When you give your blood, what matters are your antigens. So if I am type B negative, my blood just has the B antigens on them. So the only people who can't take that are people who have the anti-B antibodies. So that would be anybody in blood type O, anybody in blood type A. But everybody else can receive it. So it matters your antigens. When you receive blood, what matters are your antibodies. Your antibodies determine what you will recognize as being foreign. So notice if I have A positive blood, the only thing I recognize as foreign is the B antigen. So as long as I don't get the B antigen, I can get any type of blood. Whereas notice if I'm B negative, I have the A and the RH antibody. So I can't get anything with A in it, and I can't get anything with RH in it. So notice I can only get B negative, and I can only get O negative. Do those two rules help us to make sense of it?
Yes.
So let's take it one step further and look at these two, which hopefully will be a little bit easier. Who can, and here, let's do this. Now that I've done that, I'll move this here and this here, give myself a little more room. All right. Who can receive AB positive's blood?
AB positive.
Anyone else?
B negative, AB negative.
Can they receive his blood?
Oh, no.
No.
Yeah.
But if you were AB negative, you would have the RH antibody. So you would recognize that as being foreign and you would not. So I'll answer the question. No. All they can give to is AB positive. But whose blood can they receive?
AB positive and negative.
Oh, I have these backwards. Whoops, hold on.
I was so confused for a minute. I was like, wait, hold on.
No, my bad. That goes there.
I know I got confused. I'm like,
All right, hold on. This is getting too confusing. So let's just cheat and do that. All right, let's do this again. They can give to AB positive only. Excellent. All right. Who can they receive from?
AB positive.
They should be able to
AB
receive
negative.
from everyone, right?
Yeah.
But they don't have any antibodies.
They have literally no antibodies, so they literally can take any type of blood. These are the people who truly can pick blood off the ground and just shove it in their arm and know that it's going to be a match. Again, that doesn't mean that there are diseases and stuff in it, so don't be picking blood off the ground and stick it in your arm, but you get the idea.
Hypercyclides, they're good.
Yeah. And so again, as we mentioned, this person, the AB positive, is truly the universal receiver.
Professor, just to clarify, if they were AB negative, they could receive from all the A's and B's and the O's, except they all had to be negative, right?
Correct. Yes, if you were AB negative, you could get AB negative, B negative, A negative, and O negative. Oops. But no positive blood.
Got it.
You're correct. And let's do the same thing for good old O negative here. Who can he receive? Oops.
O negative.
Only O negative.
Right. Any other blood that we try to give them is going to have an antigen on them that they're going to recognize as foreign. However, look at how bright and shiny and clean the surface of this blood cell is. Is there anything on the surface of this blood cell that anybody's blood could grab onto and say, hey, this doesn't belong?
No.
No.
No. So as someone mentioned, truly O negative is the universal donor. So they can truly give to anybody, AB positive, AB negative, A positive, A negative, B positive, B negative, O positive and O negative. They are universal donor. That's why I made the joke earlier about no one willing to admit that they were O negative, so that if we suddenly become post-apocalyptic, those are the people we're going to hunt down and use for blood banks, right? Keep them locked up in our basements so that we have that blood available for us, and that while we deal with zombies or whatever other apocalypse is going on at that time.
But is it true that O negative, or like in general O type, there are like few of them that have this kind of type of blood?
So that's a great question. So notice, let's cheat and go back to the lecture. So we did all of this. We talked all this. Notice here we see a picture showing some of the differences in the blood type. So yes, O is the most common, making up about 45% to 50% in most populations. In Native American, there's a massive number of O, but you are correct. There is for the negative, it is much, much less. The vast majority of these are indeed O positive. So yes, there is a smaller number of them, far less than half that are O negative. It is a small population, and it's also a very valuable. If you are O negative blood and a blood bank has ever found that out about you, then you're probably getting calls at least once a month, if not more, asking you to come in and donate blood for them. All right. So last point about this before I want to move forward is there were two other really excellent questions that were asked that I want to address. The first one has to do with the RH just coming from mom. No, like all blood types, your blood type is determined by both your parents. Both your parents put genes together. But here's the problem. If good old mom is over here in the negative, and dad, when he gives his genes to the baby, let's say the baby ends up being B positive. That positive antigen in the baby's blood, especially upon birth, birthing, as many of you may be aware, is a relatively messy process. And it's not uncommon for mom and baby's blood to come in contact with each other. And so when mom's B negative blood comes in contact with baby's B positive blood, what ends up happening is mom builds up this big, huge defense against the RH factor. And then mom has a second baby, and that second baby gets the RH factor from dad. And now mom's blood, mom's immune response, mom's antibodies can actually attack, they can cross the placenta and actually attack the baby's blood and cause major issues. So it isn't so much that mom causes the RH. The problem is if mom is RH negative, then you have to be worried about what dad's blood type is, because if dad's is positive, if dad's negative, then that means that the baby's going to be negative and it's not an issue. But if a dad's positive, then knowing the blood type of the baby can be important because you can get this RH reaction. And so if mom's blood type is negative and dad's positive, what will they often do when you're pregnant?
They'll give you the, I think it's Rogam shot.
Yeah, there are a couple.
But what does that actually do?
It suppresses the production of these antibodies. So it discourages mom's body from building up that defense against the RH factor.
And I think, doesn't that have to be given by a certain point in the pregnancy? Otherwise, I just have a friend that happened to, and they waited too long and they had to stop having children after that.
Oh, wow.
Because of the risk.
Well, then you actually know the question and the answer to that more than me. I was not aware of that, but that doesn't surprise me. That again, the sooner that you need to do this, because like I said, the more mom's in contact with that blood, the more of a vigorous reaction she can have. And if she's built up a strong defense, then you're not going to be able to suppress it anymore. And then, yeah, like you said, then there's a very strong increased risk of causing damage to a positive RH baby.
Professor?
Yes.
They also say that the cause of jaundice in babies has to do with the blood type, but it doesn't make sense because like 2 of my kids had jaundice, but both my husband and I are positive. So how was that connected to that?
You are correct in that jaundice has to do with red blood cells, but it doesn't have anything to do with blood type. In fact, current studies show as much as 30% of babies show some level of jaundice. So having babies that are jaundiced is quite common. What happens is our fetal blood is very viscous, has a massive number of red blood cells in it, which kind of, if you think about it, makes sense. As we've learned, more red blood cells you have, the more oxygen carrying capacity of your blood. And if you think about it, you're probably not more metabolically active in your life than you are as a fetus growing from a tadpole into a person. So had that massive amount of metabolism, massive amount of growth, requires a massive amount of oxygen. So what happens right about the time of birth is that a large number of those red blood cells are broken down and destroyed. Why that becomes a problem is for many babies, their livers are not fully functional yet. And because their livers aren't fully functional yet, it's not able to break down all of the bilirubin, which is one of the byproducts. Here, let's actually switch slides so I don't have to mess this one up. Bilirubin is a byproduct of the breakdown of red blood cells. And normally our liver breaks it down, turns it into bile, recycles it to make more red blood cells, things along those lines. But if the liver is not fully functional yet, it can be overwhelmed by this massive amount of red blood cell destruction. And as a result of that, the baby builds up bilirubin in their blood and they take on that yellowish coloration, that jaundice. Now, jaundice in and of itself isn't a problem. As I mentioned, as many as 30% of babies have them. However, typically it follows a very standard pattern where it'll peak usually around day 2 or 3 after birth and then dramatically drop after that. So if the baby is jaundiced, they will monitor it relatively closely. If it gets too high or stays high for too long, it can become a problem because it can inhibit the development of the nervous system. So too much of it is absolutely a bad thing. Luckily, grandma knew how to handle it. How did grandma handle jaundiced babies?
Whiskey.
No, that's what you do when they cry. Right? That's what you do with colic babies is whiskey. There you go. Grandma would put them in the window. Not like a potted plant out on the windowsill, but she would have a nice diffused curtain and would put it in that indirect sunlight. Which is why if you've ever gone to a NICU, you'll see that ICU for the babies has just rows and rows of windows that they put the babies in front of, right? Okay, well, not exactly, but often grandma gets things right. And this is one of the cases, well, okay, not often. Occasionally grandma gets things right. And this is one of those cases. What was it that putting that baby in the window did that helped the baby?
UV light.
UV light. It turns out that UV light converts bilirubin into a more easily digestible form, making it easier for the liver to break it down. So yes, the NICUs don't have rows and rows of windows that they put the babies in front of, but they have plenty of UV blankets and UV lamps and other types of things that they can put the babies around or under to help to convert that bilirubin into a more easily digestible form to get rid of that excess bilirubin. So it has everything to do with red blood cells, but it has nothing to do with blood types. All right.
That's just an easy answer to your question. Like they just tell you it's your blood type.
Well, my guess is, as we've talked about in this class, I often give you guys, as I've said before, often when we talk about these things, we talk about these things as I explained to you that the sky is blue, right? When I tell you the sky is blue, I'm not lying to you. But there are plenty of cases where the sky isn't blue. At night, the sky isn't blue, sunrise and sunset, when it's raining, when California is on fire, the sky is often not blue. right? It's not that I'm lying to you. I'm just giving you a simplified answer that's kind of correct, mostly correct, but it's a simplified answer. My guess is they were trying to give you a simplified answer because most people don't appreciate these things as well. And again, with a newborn baby, the last thing you want to be worrying about is potential nervous system damage. So my guess is that they kind of were oversimplifying it a little bit when they said that. But yeah, it has nothing to do with, it has everything to do with the amount of blood and nothing to do with blood type.
Thank you.
Yep. All right. So that was one of the questions that was asked. The other question that was asked that I want to address now is someone mentioned, how do you know? How do you determine blood type? Anyone know how this is done?
Well, if you paid attention during Labster, I'm guessing that
There you go. Excellent. All right. So perfect. You got the right idea. Absolutely. What happens is you've got these 3 wells on your tray, and this is something that we normally do in the classroom, and you fill them with a sample of your blood. So this is some unknown blood that we don't know what it is. Then to each of these, you are going to add the antibodies. So in this case, I will add the known anti-A antibody. To this one, I will add the anti-B antibody. And to this one, I will add the anti-RH. What we are looking for here is a process known as agglutination. We have these red blood cells that have an antigen on them. And not just one, but as we've talked about, they have hundreds of these A antigens on their surface. Were the A's the diamonds? I don't remember now which one I made the diamonds. Doesn't matter. Was it A?
B.
Did I make A circles?
It was B.
Yeah.
Okay, B was the triangles. All right. All right, B. So excellent. So I have blood type B here. And so when I add that anti-B antibody to it, remember, as I mentioned, it has 2 binding sites. So it can bind to this red blood cell, but it also has a second binding site that would allow it to bind to a second. And then another antibody would be able to bind that to a third, and another antibody would be able to bind that to a fourth. And notice, just that simply, we get a clumping of the blood together. Now, if this was happening in your blood vessel, would this be a good thing?
No.
No, this would be a horrible thing. So in this case, this agglutination is the antibodies cause a clumping of the blood. That is why we want to be very careful about what kind of blood we put in our body. But we can use this to our advantage. So let's say, for instance, in this particular case, when I mix my A antibody in with this blood, what happens is it starts to clump all together. We get that agglutination that occurs. What does that tell me about my unknown blood cell?
It definitely has A.
It has A, which you told me I was circles. Excellent. So we know it has A because it clumped with it. And then I mix the B antibody in it and it clumps again. And what does that tell me?
No, the blood type is AB.
Well, we're not done yet, but you are. So what is the one piece of information this tells me?
positive for B
Positive for
antigen.
B.
Yeah, it means that my unknown blood cell has the B antigen on its surface. I then mix the RH antibody into it and nothing happens.
So
What
it's
does
negative
that
for
tell me?
RH factor.
A, B negative.
Exactly. So that means that my cell does not have the RH factor antigen. So notice each one of these three tests gives me one single piece of information. But as you pointed out, when I then put those three pieces of information together, do I know the blood type of this unknown person?
Yep.
Yes.
What would it be?
A, B negative.
There you go. Oops. So by looking for agglutination or the lack of agglutination, we can determine the blood types. Remember, each test just tells us one thing. This first test told us that the cell has A antigens. The second test just told us that the cell has B antigens. The third test told us that the cell does not have the RH antigen. And then when I put all three of those pieces of information together, I have my blood type. So that answered, I think, the last question that somebody asked about how do you determine the blood type.
Professor, how long does it take for clumping to occur?
It happens in less than 30 seconds. Basically, you add the antibodies serum, and then you agitate it with your stirring stick or toothpick or something like that. Usually the reaction takes less than 15 seconds. It may take as long as 20 or 30, but definitely less than a minute. It's a very rapid test.
Got it. So if there was a doctor and they put the wrong blood in the wrong person, they would immediately see a reaction?
Yes, absolutely, yes. Yeah, like I said, if we were in the classroom, that's cool that Eric, that you got to do it. And like I said, if we were in the classroom, you guys would actually be doing this as well. They don't let us work with real blood, so it would have been fake blood, but you would have gotten a chance to be able to do this test, absolutely. I'm sorry, I spoke over someone. Did someone else say something?
I was just gonna say, because it happened so fast, if on the rare occasion that someone did get the wrong blood, could they stop it fast enough before it had like catastrophic results? Or
so here's the problem. The second you put it in, you're going to start causing a clumping of their blood, and we know how fast the blood moves through their body. The problem is, by the time you start to see a reaction in them, you can stop what's going in, but there's not much you can do about what's in there. I mean, basically, they will try to use dialysis to get the clumps out. So, I mean, they will try to do it as fast as possible, but in all likelihood, you're going to definitely have some serious problems as a result of that. Absolutely. So, because by the time you're aware that you've made a mistake, you're aware because some damage has already been done.
But this will cause their life, right? It could possibly be a reason.
In an extreme case, yes, it could cause someone to die, because notice, if you clump blood like this together, is it going to be able to pass through capillaries?
no?
No, so you're going to start to get blockage. And if that blockage occurs in places like your brain or in places like your lungs or things like that, it can cause severe, severe issues. Yep.
I have one more question about the pregnancy.
Yes.
Really fast. This just made me kind of brought up a question. So I feel like because it's such a big deal, the RH factor with pregnancy, what if you don't know the father's blood type, or in a situation where you don't know, can you tell through a blood sample from the female? Or if not, would you just give the RhoGAM shot? I just feel like no one talks about it, and it's kind of could be a big deal.
So obviously, the first thing you're going to want to do is check the blood type of the mom. If mom has a positive RH, then everything else is a moot point.
Right.
Yeah, that's what happens in our culture. We actually do the blood test before marriage.
Yeah, and so that's not uncommon. If there's a situation where a female is pregnant and doesn't know who the father is, or for whatever reason the father is unwilling to give a blood sample for the testing, then by testing the amniotic fluid, they can determine the blood type of the baby. It's not ideal to do that. Obviously, anytime you're putting a needle into the womb for any reason, there's always risks involved in that. But if mom was negative and dad was unable or refused to provide that information, then they could ascertain it from the baby themselves. If there was some risk as to taking, the amniotic fluid, then my guess is, yeah, they probably would just give the Rogram as preventatively. But, I'd like to say that hopefully a woman isn't, having kids with a person who's not willing to at least give them their blood type, but
I guess more so I just don't like hear of, like, I don't have children, but I don't like hear my friends say, oh yeah, I'm pregnant and my husband's going in for this blood test. Like, I feel like it's just an important thing but maybe I just haven't had.
Well, but at the same time, at the same time, many people know their blood types, either because they've had surgeries or they've had injuries or they've donated blood. So, a lot of people already know a lot of people already know their blood types to begin with. So then my guess is that's probably more it. Yeah.
Thank you. Sorry, I was just kind of, my brain was just going.
Definitely, like I said, in that type of hopefully theoretical situation, you can get that information from the amniotic fluid. So that would be one route to go. Or like I said, not my area of expertise, so I don't know what the potential risks are for ROGAM. If there are very few risks, then my guess is that yeah, you could just give that preventatively. But I'm not familiar enough with the medication to know what kind of risks are involved with that, where if that'd be something you would just want to take regardless. So that I can't answer.
Thank you.
All righty. That is the last bit of information that I was concerned about or wanted to cover for today. Again, as someone pointed out, this is similar to what you were doing with your lab simulations. So hopefully this is not completely unfamiliar information, but I think it's worthwhile to take the time to make sure that we understand these kinds of concepts. So that is everything that you are going to be responsible for on the next exam. So what we will do now is we will go ahead and take one more break. So anybody whose brains are full and they want to totally be done and leave, they're welcome to do that. However, when we come back from the break, then I will happily answer any questions you guys have, either about specific content for the exam or the mechanics of the exam itself, the logistics of how it works. So again, for anybody who didn't have me for 4:30, if you have questions about the process of taking the exam, I will happily answer those as well. So we went a little long on this last part, so let's go ahead and, well, it's not really a little bit longer. Let's meet back at 3 o'clock. So we will come back at 3. And at 3 o'clock, we will then start the recording and do our questionnaire review. All right. Any questions on any of that before we take our break? All right. I'll see you guys in 15 minutes, 16. You get an extra minute. All right. See you guys at 3. So as I mentioned, this point, we are done with all of the new material you're going to be responsible for on your exam for Tuesday. So now this is your opportunity to ask questions of me, clarification on the process of the exam or any of the information for the exam, whatever I can do to help you to be successful come Tuesday. So this is the point where you ask the questions.
Could you go over just quickly, like the 3 different circulations?
Sure. So let's start easy. Let's just identify them by name. What are the 3 circulations?
Systemic.
Excellent.
Pulmonary.
and
Coronary.
excellent. Now that we've identified them, we can actually identify what they do. Obviously, all of them are involved in blood. So this is blood that starts in the heart and it goes to what?
The body, like the body's
Body.
systems.
Yeah. Tissues of the body. And then where?
The superior, inferior vena cava.
True. Oh, excellent. Which goes back to the heart. Exactly. Excellent. And that's whereas pulmonary is what?
Lungs.
Vertical lungs, back to the heart.
And coronary is
Heart to heart.
Heart to heart.
let's make it heart walls just to be different. Excellent. But I like that other bit of information you were talking about as well. Let's at least talk about what great vessels or chambers are involved. So in this case, the systemic circulation begins where?
Left ventricle.
Right.
Excellent. And from there goes into
Ascending.
Ascending aorta.
where it then takes its merry path and it comes back via what? Someone mentioned the,
Vena cavas.
yeah, the inferior and superior vena cavas. And then into,
Right atrium.
Right atrium.
how does that relate to our pulmonary circulation?
It starts at the right ventricle. With the pulmonary trunk. Then the left and right pulmonary arteries.
Right, from the trunk to the arteries. I like that. Yeah, and since the trunk's small, we can do that.
And then comes back to the left and right pulmonary veins into the left atrium.
There's one other major difference between the systemic and the pulmonary circulations. What is it?
Oxygen rich and oxygen poor blood.
Excellent. So in our systemic circulation, arteries carry what?
Oxygen rich.
And our veins?
Oxygen poor.
Excellent. And it looks like I'm gonna have to make this whole thing smaller so it all fits. Perfect. Whereas our pulmonary circulation, arteries carry
Oxygen poor.
which means that the veins carry
Oxygen rich
blood. Right. In fact, it's that pulmonary circulation that screws it up for us when we want to try to identify blood vessels by their oxygen carrying condition. Most arteries and veins are systemic, so most arteries are oxygen rich and most veins are oxygen poor, but most isn't all. So we want to make sure that when we define them, we use characteristics that are true for all of them. And all arteries carry blood away, all veins carry blood back to the heart. Excellent. So that brings us to our coronary circulation. Where does it start?
Left ventricle.
Okay, it also is going to start in the left ventricle. From there into what?
The ascending aorta.
Which makes it similar to what we saw in the systemic. However, in the systemic, from the ascending aorta, it went into the aortic arch. Is the same thing true here for our coronary circulation?
No.
No.
No, where does it go here?
The left and right coronary arteries.
Excellent. And where does it end?
The right atrium,
True, it's going to go into the right atrium from what?
coronary sinus.
There you go. Excellent. And since we did it for the others, let's do the same thing here as well. Arteries and veins. What is the condition of the arteries and the veins in our coronary circulation?
Oxygen rich.
And that makes our veins oxygen poor. Excellent. Is that kind of what you were looking for?
Yeah, thank you.
Excellent. Excellent. Any other questions on this? I see your question, Danielle. Let's make sure there's nothing else on these circulations and then we can switch gears. So any other questions on this? All right, perfect. So again, one of the things that I will say to, again, beat this horse dead, people often lose points, not because they don't know the information, but because they don't read the questions carefully, and because you got to be very, very careful with your verbiage, both the way that I describe things of what I want and how you describe things to me. One of the things you want to be careful about is mixing terms together. I can see what you're interested in is understanding more about the cardiac cycle, but be careful, because remember, the isovolumetric contraction is only one part of the cardiac cycle, and it's not even the first part. So rather than all of the elaborate drawings and all the things we did last time, why don't we just identify the steps? What are all the steps in the cardiac cycle? Where does, and again, it is a cycle, so it is a continuous process, but in this case, it is a continuous process that has a fairly appropriate starting point. So what did we use as our starting point for the cardiac cycle?
Ventricle filling.
Okay.
Quick question, Professor.
Yeah.
On the exam, which I'm sure I have a good feeling you might ask us a question like this. Do you want us to actually go like 1, 2, 3, 4, 5, and then name each thing like ventricle filling, ventricular systole, or can we just start off with 5 and then name everything that's going on in there? Or do we have to give a title to each section?
So
kind of like how this
okay,
is.
so I'll explain my hesitation. In most instances, what I care the most about is the detail, the information.
Mm-hmm.
Okay. However, the problem with the cardiac cycle is we have things like isovolumetric contraction, right? And so that term definitely has some significant meaning to it.
Mm-hmm.
Now, if you did a perfect job of explaining exactly what happened and how the pressure is changing and the volume isn't and why that's occurring and all that, you likely would get full credit. But honestly, it's hard for me to envision explaining that process without giving that name.
Mm-hmm.
And even here, like we were talking about here, again, obviously the detail is more important, but here's part of the problem. The first step is ventricular filling, but even that, is that really just one thing?
No.
No. What is that? How many things really is ventricular filling?
A couple.
2 things.
2 things. Excellent. And how would you differentiate those 2 things?
Passive versus active.
Yeah, exactly. So first we have a passive ventricular filling, and then we have an active. Again, you probably could get away without using those terms as long as you described everything that was going on in them.
Mm-hmm.
But my hesitation was just when we're talking about very specific events that are occurring, it's hard to be completely descriptive in those events without actually naming them.
Got it. Okay.
Does that make sense?
Yeah.
But would it be possible to give me a 100% correct answer and never have the term isovolumetric contraction in it? Probably. It would, as long as it had all the information, all the details, but I would be surprised by it. But again, I wouldn't ding you not for having that term as long as you explained the term fully, explained what fully was happening during that stage. Then you wouldn't get dinged not for just not having that term. But if you didn't have the term and didn't fully explain it, then you might get dinged there.
Got
Does
it.
that make sense?
Thank you.
Excellent. No, it's a great question. And again, I wanted to give it an accurate answer. That was why my hesitation with that, because it's not because you're right. A lot of times, like, and again, I think back to like 430, when we're talking about endochondrial ossification, right? Saying that, the formation of the primary ossification center, you didn't necessarily have to have that named as long as you're describing what is occurring during that event. So labels aren't always important, but they can be helpful in describing. But yeah, I'm definitely more interested in information. All right, speaking of information, obviously what's happening during both active and passive ventricular filling.
The ventricles are being filled.
Yeah, there you go. The ventricles are being filled in blood. So therefore, what is the big difference between these two?
Whether the atria is contracting.
Excellent. And so obviously during the active, this is when the atria plural, are contracting and they're forcing blood into the ventricle. Whereas at this point, the atria are relaxed. In fact, there's no muscle activity. It is just pretty much gravity and fluid pressures that are moving the blood into the ventricle. And remind me again, which of these two is more involved, or which of these two put more blood into the ventricle?
Passive.
Yeah. Why do you think that is? Because it is a more powerful process?
No, mainly
No, it's
gravity.
just-
Okay, gravity, but why else might more blood enter during the passive ventricular filling stage?
Is it because
The ventricles are empty,
That's
almost
part.
empty.
Yeah. All right. Let's ask the question this way. Do you think that the atria are equal in the time that they are relaxed as to when they're contracted?
no, there's more volume when they're open.
Yeah, not only is there more volume, but this is a longer period of time. It may be a slower dribble, but right, if you've got a slow leak in your sink, but you leave it on for 5 days, right, your sink's going to flood. So it may be a slower process, but it's a longer process. And so it gets more blood in there. All right. So those are our first two phases. What happens next? What happens after our ventricles are filled?
They can
Ventricular systole.
Okay, so you get the right idea. We get the start of ventricular contractions. And again, yes, the systole. Why is that significant? What's the first event that happens? The AV valves have been open, which is why our ventricle has been filling. But what happens when our ventricle starts to contract?
They close.
All right. What's the state of the SL valves? And don't use AV and SL on the exam. Remember, spelling matters. On an essay question like this, if you want to write it out once and then abbreviate it after that, that's fine. If you want to have a key at the top or the bottom, AV means atrioventricular or something like that, that's fine. Make sure you just spell it out once. And then, well, I wasn't actually commenting on you, Samuel, I was more counting on me. But just to remind you, don't fall into the errors that I'm falling. So make sure that you're doing that. Excellent. So atrioventricular valves closed from the start of the contraction. What was the state of the SL, the semilunar valves prior to this?
Closed.
were already closed. And so now all the doors are closed. Why is that significant?
You have your end diastolic volume.
Yes. No new blood can get in to the ventricle. It has reached its max volume. And what did we call that volume again? I know you said it, but say it again.
The end diastolic volume.
Excellent. Perfect. That is a good start. But something else is happening as these ventricles are starting to contract as well. The doors close and that don't change right away, but what is changing?
Does the atria relax?
True, but is that really affecting the movement of blood in this case?
No.
No.
Pressure
Excellent.
increase.
There you go. As the ventricles continue to contract, pressure increases. So notice here, our pressure is increasing inside the ventricle, but our volume has stayed the same. Is there something we might refer to this particular stage as?
Isovolumetric contraction phase.
Excellent. This is when that isovolumetric contraction is taking place. The pressure is getting greater and greater, but the volume isn't changing. It's like me holding a can of soda. I can hold that can of soda and I can squeeze as hard as I want. And as young and vibrant and healthy as I am I going to be able to crush that full can, pop at the top off of it and spray the soda up into the sky?
No.
No, of course not. So I can squeeze as hard as I want and the volume's not going to change. And even as I get to squeeze it more and more and more and more, it doesn't. However, if I put it in a vise instead of just in my hand, can eventually I get that pressure great enough to pop the top?
Yeah.
Yeah. And isn't that what happens to our heart?
Yep.
We build up enough pressure in the ventricle where it becomes greater than what?
Pressure in the arteries.
And when the pressure in the ventricle is greater than the pressure in the arteries, what happens?
The semilunar valves open.
Excellent. And what event takes place?
Ventricular ejection.
True, that's what we could call this. So again, if we wanted to give a name to this state, to this stage, we could call it ventricular.
The contraction of the ventricles.
But yeah, absolutely. When those semilunar valves open, blood exits the ventricle into the arteries, right? So notice we can give it a name, ventricular ejection, or we could also describe what exactly is happening. This kind of gets back to that question you were mentioning before. On the exam, if you put step 4, ventricular ejection, and then left that there, or if you put step 4, pressure in the ventricles become greater than the pressure in the arteries, the semilunar valves open, and blood exits the ventricles into the artery, which of those two statements is going to get you more points on the exam?
The left.
Yeah. So notice we didn't have to say ventricular ejection here. And just saying ventricular ejection is not the magic term that's going to get you all the points. I'm much more caring about the description. Again, my hesitation was all about this one and the next one because they're such important terms. And that's a perfect thing to lead into what we were going to talk about next. Is that ventricle going to keep producing massive amounts of pressure forever?
No.
It's going to start to relax and the pressure drops. What happens when the pressure drops below the pressure in the artery?
The blood starts to backflow.
Tries to backflow, but is it going to get into the ventricle?
No, because the semilunar valves are going to close.
Notice once again, all four, that's not how you spell four, valves are closed. So there's no change in, no more change, let's say it that way. No more blood can leave the ventricle. What do we call that?
And systolic volume?
Excellent. While all four doors are closed and the volume doesn't change, does our pressure change? All right, I'll ask the question a different way. What happens to our pressure?
Does the pressure drop?
Yeah. As the ventricle relaxes more and more, as the ventricle relaxes, the pressure drops more and more and more. So notice again, we're at this state where pressure is decreasing and the volume stays the same. And do we have a fancy term for that?
Isovolumetric relaxation.
However, do all four doors stay open forever? I mean, pardon, stay closed forever?
No,
No, eventually what happens?
you go back to the beginning of the cycle
How?
by having the atrioventricular valves open again.
Excellent. And how do they open again?
Passively.
True. What causes them to open?
Oh, the pressure of the atria is greater than the ventricles.
Pressure in the ventricle drops below the pressure in the atria. Notice the atria aren't contracting yet, but that atrioventricular valve has been closed and they've been slowly filling with blood. And as the amount of blood in the atria increases, there's going to be a higher pressure in the atria. So as the pressure of the ventricle drops, and when it gets below the pressure of the atria, that's when our AV valves open. And we start the passive filling of the ventricle. And as you so concisely put it, that puts us right back at the beginning again. Questions on this?
Professor?
Mhm.
So this would follow the ECG assignment then? The like the interval and the segments?
So great question. Remember, the electrical activity of the heart, which we measure with the ECG, is related to the mechanical activities. So if you remember on that assignment, we talked about the mechanical and electrical events. All we've talked about here are the mechanical events, the contraction or relaxation of chambers, the opening or closing of valves. Those are the mechanical events. The ECG measures the cardiac conduction system, which are the electrical events, the production of the action potential, the spread of the action potential across the atria. Now we know the electrical events are going to lead to the mechanical events. We know when you stimulate the atria are going to contract. However, are those electrical events, is that ECG a direct measure of these mechanical events? For instance, if we measure the size of the P wave, can we tell how long the atria is going to contract for?
No.
No. So it's not a direct measure of the mechanical events. We know one leads to the other. When a cell is excited, it's going to contract. When the cell relaxes, it's going to, or repolarizes, it's going to relax. So we know the the electrical events lead to the mechanical events, but it's not a direct measure. So the goal in that activity was to describe the electrical events as they were occurring, and then relate that to what type of effects it might have on the heart mechanically.
Okay, thank you.
Cardiac cycle is really just the mechanical events. Great question. Any others? Anything else on the cardiac cycle?
Could you talk about the cardiac conduction system and its modifications with the parasympathetic and sympathetic system?
Sure. Okay. So again, there are 5 components to the cardiac conduction system. What are they?
The sinoatrial node, the atrioventricle node, the atrioventricular bundle, the bundle branches, and the Purkinje fibers.
Let's do this. Rather than going through all the process, let's talk about what each of these things do and how they can. So let's answer your question. Sinoatrial node, what is its function?
Generates action potential.
Okay, generates the action potential. Although really any of these can generate an action potential. So what's so special about this action potential?
Pacemaker.
Yeah, this is the action potential that really drives the activity of the heart, right? We could use that fancy word pacemaker, or we could say what that pacemaker does. It basically sets the heart rate. Okay. So this is where we're setting our heart rate. So not surprisingly, this can be also where we can modify it. So what effect, so what is our sinoatrial node? So as we know, there are 2 branches. Is it innervated by both the sympathetic and the parasympathetic nervous systems?
Yes.
And what does the sympathetic nervous system do to the sinoatrial node?
It
Excites it.
increases
Okay, how?
heart rate.
How does it increase that firing rate?
By increasing the amount of action potentials it generates.
Okay, how?
The release of epinephrine.
Excellent. Which open norepinephrine, really, which open sodium ions. Excellent. Primarily norepinephrine, but also epinephrine as well, which opens sodium channels, which increases the firing rate. Conversely, what does our parasympathetic nervous system do here?
Releases acetylcholine.
Which opens,
Is it chloride channels?
yep. And that decreases, if I remember. Excellent. What does the atrioventricular node do? Excellent, I like that. How does it do that again?
Has cells that are small and like thin, so it I think increases permeability, and so that decreases.
It's not so much permeability. Remember, permeability would let more things in, go through it faster, so that would actually make a bigger signal. Remember, it slows the signal down by having a high resistance. Remember, resistance is the opposite of flow. So if it's harder for it to flow through there, if there's more resistance, it's going to move slower.
That because it has less gap junctions?
No, remember, the axons of the cells themselves are very thin. So the action potential moves very slowly through it because it has such a high. Remember, if you think back to 430, when we talked about propagation of an action potential, remember, it was primarily sodium moving down the axon, which then opened more voltage gated sodium channels, allowing more to come in and more move. So if we can slow the spread of the sodium down the action potential, then, I mean, down the axon, then we can slow down the action potentials. And notice, just like before, it is also innervated by both the sympathetic and the parasympathetic with the same types of effects. So we can either increase the speed or decrease the speed. All right. So basically, we're not going to go bother going through that whole thing again because that's pretty much the same. Let's also simplify things a little bit by throwing the AV bundle and the bundle branches together. Collectively, what do they do?
Propagate the action potential down to the apex.
So within that wall of the muscle between the two ventricles, it carries that action potential down to the apex. Are either of them really innervated by either the sympathetic or the parasympathetic nervous system?
Only the sympathetic.
So really, it's a great, it's a good thought, but remember, this is deep inside of the heart. Remember, as we saw, the sympathetic connects to the Purkinje fibers, right, which are more superficial. Neither of these are really innervated or affected by that, because basically we want it to reach the apex of the heart. So this part really isn't modified in that way. Where we actually run into problems is damage. If we have damage to the system, especially to the singular bundle branches, this is a condition we actually call heart block, where that heart block can actually slow the signal or delay the signal or actually cause signals to fail to reach the apex, in which case they wouldn't allow the ventricle to contract. So damage in this area, it's a vulnerable part of the heart, but it isn't something that's necessarily innervated by the autonomic nervous system. However, you are correct. The Purkinje fibers, these carry the signal up the ventricles. Again, this gives us our effective directional contraction pump. But as you pointed out, this one is innervated by the sympathetic only. Again, it releases norepinephrine. It opens those sodium channels, but what's the effect? What does the sympathetic nervous system actually cause the Purkinje fibers to do?
Increased contraction.
Yeah, basically they produce a bigger signal. And with a bigger signal, we get a stronger contraction. So Jacob, did that answer your question?
Yes, thank you.
Excellent. All right, questions on this? All right. What's next? Oh, wait, hold on. There was a question. Yes, great question. So when the action potential reaches the apex and then starts to spread up the ventricles, that is what stimulates the walls of the ventricles, causing the ventricular contraction. That is correct. And so again, by traveling from the apex up, it starts the contraction at the bottom and gives us that nice effective squeezing motion. Because remember, the great arteries that we want to pump into, the ascending aorta and the pulmonary trunk, are at the top of the heart. So we get a directional pump towards the top of the heart, making that pump very efficient. Any other questions on this one? All right. What's next?
Professor, could you go over a bit more in depth on venoconstriction? There was a question on the daily quiz today, and I was kind of confused on it.
I don't remember the question. Do you remember what it was?
It was something along the lines of like venoconstriction either increases or decreases or something, which I'm not sure.
Okay. So when we talk about venous constriction, what are we talking about?
Constricting the vein.
Right, exactly. Remember, as we talked about, it is the differences in the pressure gradient that cause blood to move. And remember, as we saw, the veins have the smallest pressure gradient from the start to the finish. So blood moves very slowly through the veins. If you remember, there was that chart that showed that about 60% of your entire blood volume at any one time is located just in the veins. Right? Remember, it was like 1.5% in the capillaries and 8% in the heart or something like that,
Right.
Some in the pulmonary. But by far, the largest amount was in those veins. So our veins carry this slow moving blood and have the vast majority of the blood in it, which, while you're sitting here calmly in your chair like I am, is totally fine. However, when you want to be active, when you decide to get up and exercise, or you have to run away from that bear with an axe, we need to get that blood moving. We need to get it to the heart. We need to get it to the lungs. We need to get it to the muscles. We need to get it to the liver so that it can get the nutrients that it needs, and get it to those muscles so they can get their oxygen. They can move and do that. So what happens at that time is we have our veins constrict. And so when those veins constrict, that pushes the blood faster back towards the heart. Basically, it's like turning the faucet up. And so as we've talked about, one of the things we talked about with cardiac output, how much blood you can pump out of the heart is, definitely is affected by how much blood you can get into it. So when we constrict those veins, blood moves back faster to the heart. So faster moving blood is going to fill the heart more. And remember, we also talked about that Frank-Starling effect, where the more the heart expands, the stronger the contraction it's going to produce. So that venoconstriction helps us to get the blood going to the areas that we need in those times of stress, those times of activity, and it helps to fill the heart more rapidly, get more volume in there, getting it to contract more strongly, which is going to increase our stroke volume. And of course, if you increase your stroke volume, you're going to increase your cardiac output. And so we can get the blood moving to more areas more quickly by constricting those veins.
Got it. Okay. Thank you.
Yep. What's next? Sure. Let's actually do it without numbers this time. We can figure out the math later. So when we talk about capillary, there are basically two types of forces. Those forces that are out of the capillary and those forces that are into the capillary. Oops. Oh, I did this wrong all sorts of ways. Excellent. How many total forces are involved?
4.
Two types. Excellent. What are the two outward forces?
Was it the hydrostatic pressure on the capillaries?
Yep, I spelled pressure wrong. Excellent. The force of the blood against the walls of the capillary, trying to, leak things out. And what else? Osmotic pressure of what?
Interstitial fluid.
Excellent. The osmotic pressure of the interstitial fluid, right? Things draw water to it. So the stuff in the interstitial fluid draws water out of the capillary. There you go. And hopefully if we figured those out, then it should be pretty easy to figure out what the two inward forces are. Give me one of them.
Osmotic pressure of the interstitial fluid.
Excellent. No, not of the interstitial fluid. Osmotic pressure of what?
The blood.
Again, stuff draws water to it. In this case, it's the stuff in the blood that is pulling water into the capillary.
Oh,
And
sorry,
so
the
it's-
osmotic pressure of the capillary.
True, of the capillary of the blood. We could call it either way. The osmotic pressure of the capillary, because really it's the blood. It is in the capillary, but it is the blood that has the stuff that is drawing the water to it. So I certainly wouldn't mark you wrong for saying it's the osmotic pressure of the capillary, because you've got the right idea. But if you think about it, it's really the blood that has the stuff in it that is drawing it towards it. And so then what's our last into the capillary force?
Hydrostatic pressure of the interstitial fluid.
Which is against the wall of the capillary, trying to force water into the capillary. So there you go. Two inward forces and two outward forces. And so we have to see how these are competing against each other. So if in a capillary, we want to know what direction water, and after all, since water has lots of stuff dissolved in it, if we want to know what direction water and the stuff in the water is going to do, we have to do some math. There are three calculations we have to make. What's the first calculation?
Net osmotic pressure.
Excellent. And how would we do something like that?
Osmotic pressure of the blood minus the osmotic pressure of the interstitial fluid.
Excellent. And let's think about this. If this is a positive number, what does that mean?
Outward force.
Well, if it's a positive
Inward.
number, is the osmotic pressure of the blood greater, or is the osmotic pressure of the interstitial fluid?
Blood.
Blood.
Yeah. And if the osmotic pressure of the blood is bigger, which way is water going to move?
Into the blood.
Into the blood or into the capillary. Excellent. What's the next calculation we have to do?
Net hydrostatic pressure.
And how do we calculate that?
The hydrostatic pressure on the arterial end minus the hydrostatic pressure of the interstitial fluid.
Again, we don't care about what end. We just need to know it's the hydrostatic pressure of the capillary minus the hydrostatic pressure of the interstitial fluid. Excellent. Right. We did that calculation twice, once at the arterial end, once at the venous end, but we also could have done it in the middle. We could also do it in a different capillary someplace else. So all we care about is that it's the hydrostatic pressure of the capillary minus the hydrostatic pressure of the interstitial fluid. Notice again, a positive number here means that the hydrostatic pressure of the capillary is bigger. And what direction would that be?
Out of the capillary.
And now that we have these two, we need to put them together. And how do we put them together?
The net filtration pressure. You take the net hydrostatic pressure minus the net osmotic pressure.
Excellent. And in this case, obviously a positive number means what?
Outward force.
And when water and stuff leaves, what do we call that?
Filtration.
Whereas if it's a negative number, that means an inward force. And what do we call that?
Reabsorption.
There you go. As I said, once you understand all of this, I can give you any 4 random numbers, and you should be able to calculate the net filtration pressure. In fact, I think I hinted pretty heavily that that is very likely to be an essay question. All right. Questions on this? All right. Any more? I can always tell when the brains are starting to get full because the questions start to slow down. But we still have a little bit of time left. I'm happy to answer a couple more if you guys have them. I will remind you that I have office hours on Monday. I'm also, especially on a exam weekend, I'm very good at checking my emails. So again, if you don't have a question now, but something comes up between now and then by all means, email me and I will happily answer those questions for you. But like I said, if you do still have more questions, I will also happily answer them now as well.
Professor, just to double check, you said that the exam will open up exactly at our class time, correct?
So yes, so the exams, both exams will be available starting at noon on Tuesday. You don't have to start at noon, but like I said, I encourage you to start close to that time And I encourage you to start with the lab exam because those are the ones that most people have problems with. But if you are one of those people who are worried about how much time it will take for the essay questions, then you could start with the lecture exam first, get that done and see how much time you have left and feel comfortable with it that way. You should have enough time to easily complete both exams and then also even take a break in between them. But yes, they will not be available until noon, and then they will not be available anymore after 4:35.
Gotcha.
Will they be in the modules or the quizzes tab?
Quizzes, they will be in the quizzes tab.
Professor, just to clarify, going back to the leukocytes, you said that the monocytes can be identified by its U-shaped nucleus, right?
Yes, although that can be tricky. What I think is more obvious about them is their size. Now, when you're just looking at them by themselves, it's not as obvious, but they are dramatically bigger than any of the other leukocytes. So their large size, their lack of large distinct granules, and they kind of have a folded nucleus. Those are the three ways that you can best identify a monocyte.
Okay, thank you.
Yep.
And then I do have a question. So for a Friday, the lab will be open, right?
Yes. So Open Lab is available tomorrow. Absolutely. Some of you may have run into an issue with the link. The link has been fixed. So for those of you who want to go virtually, you can do that. So you can go in person or you can go virtually. Just make sure, whichever way you go, make sure you sign in with Jeff and then sign out as well so that you get the credit for that.
Okay, and then do I find it on the front of the page for the home page? That's what I find the meeting, right?
So so here, let's do it this way. In our pages, not our home page, but in our pages is a list. So let me get to the right place. Come on. So there's two easy ways to get to it. If you go to the pages and view all the pages, then there's the Open Lab information here. Or the other way you can get to it is through our modules under the study tools. There is a link, so Open Lab information. So when you click on this, so here is the link that you would click to go to the Open Lab. This is his classroom, so you would open that and that would open it there. Or if you go on campus, he's in room 418. So 418 is where you enter in to go in. It's from 10:30 to 8:30.
Okay, sounds good. I just missed where I can find this. That's why. Thank you.
So those are the two easiest ways to find it. What are the... Well, so again, I think those three that we just did, hold on. Right, your net osmotic pressure, your net hydrostatic pressure, and your net filtration pressure. Those are definitely equations you need to know. I don't think there's any other equations we've talked about in this class so far. Anybody think of any other equations off the top of their head that we needed to know?
I think, what about cardiac output, heart rate times?
Oh, yeah, there you go. That's good. Absolutely. Cardiac output was one, mean arterial pressure, pulse pressure. There you go. So those are definitely ones. And so then also from that token, you also need to figure out stroke volume. So yeah, those are definitely equation you needed to go. That resistance one, remember I told you you didn't have to memorize that equation. I just wanted you to appreciate by looking at the number of it, how even a very small change in the diameter of a blood vessel can have a huge effect on the resistance and therefore have a huge effect on the flow. So you don't need to know that resistance equation. Certainly, is it useful to understand that basically flow is inversely proportional to the resistance? Yeah, I mean, that was kind of one of those things that we talked about, but I mean, it's not like I'm going to give you a number change for the resistance and you're gonna have to figure out the change for the flow. So I mean, I don't see anything like that. We talked about how that affects where we're directing the blood, but no, I don't think there's any, I don't think that's an equation you need to worry about. These definitely are ones that we talked about. So yes, thank you for reminding me about these. I knew I was forgetting something I couldn't think of. Are there any more? I think that's it then. All right, don't take my word for it. Look back at the lectures. There may be one or two more, but I think that might be it for the equations. All right, any more questions?
Where do babies come from?
Well, when a mommy and a daddy love each other very much, they put their puzzle pieces together, and those puzzle pieces form a baby. At least that's how I explained it to my daughter. All right, anything else in the cardiovascular system? All right, well then, as I said, again, read the questions carefully. People often lose points, not because they don't know the information, but because they don't read the questions carefully. Read the questions carefully so you can answer them correctly. Like I said, I will be available by email on the day of the exam, although if you're having problems with the exam, the first people you should go to is Proctorio, not to me. There's that little help window. They're much more, they can do much more in the Proctorio system than I can. I have very limited abilities to modify and change things. So they're the people that you should go through first. If you have any questions between now and the exam, then by all means reach out. Remember, I have office hours from 12:30 to 1:30 on Monday. So I'll definitely be online then, but I'm also available by email if you guys have anything else. Study hard, make sure you're here on Thursday next week as well, so that we can move on to the next part and form our groups. I know the idea of a group presentation is scary, but really it's really fun. Most people enjoy it. It's very low key, and I'm excited to see what you guys do. So it should be awesome. So I wish you the best of luck. Like I said, if everything goes well, I won't hear from you. You won't hear from me on Tuesday. So good luck on your exams, and have a great weekend studying hard.
Thank you.
All right, bye.